professor at IMT Lucca, and I have a long collaboration with Andrea Gabrielli, also Federico Giove, and the group of Marbilab. I came from Apicolato. Okay, I come from essentially I did my PhD in Marbilab. And I will talk you about today some results we found by investigating the functional connectivity of the brain in terms of its network configuration in healthy subjects and schizophrenia patients, and the approach we used to filter out information thanks to topological consideration. We know that the brain can be considered a paradigmatic example of a complex system because its functioning can be described at different spatial scales as a system of interrupting agents, and in general, the interactions among different portions of the brain at different spatial scales provides a set of rich and stable configurations, hierarchical stable configurations. This, from a point of view, from a spatial point of view. But if we focus on the dynamics that happened in the brain, we can recognize a great heterogeneity at different temporal scales. All this stuff allowed research in the past to elaborate several approaches to define the connectivity within the brain. You can have the structural connectivity, the real connections between brain regions, or functional connectivity as the covariance of a neurophysiological signal or hemodynamic signals. Okay, here we will focus on the hemodynamics of the brain, the hemodynamics activity of the brain as measured by Bolt signal, and we did it in the resting condition. Why we choose the resting state as a possible proxy of brain activity, cognitive activity, or brain functioning. We know that neurons in the brain operate under a balance of excitation and inhibition to maintain the neural microcircuits under a proper operational range. Moreover, further, we know that neuronal communication is sustained by the between groups of populations of neurons is sustained by the coherence between activity between among the groups, and the absence of this coherence can prevent communication between groups. Finally, we know that the level correspondence.
Can you explain what is a neuronal coherence? What do you mean?
Neuronal coherence is the coherent activity, so the synchronization of the activity among different neuronal groups. And we know also that the level of correspondence between the energetics of the brain, which is the cerebral glucose metabolism, and the synchronous fluctuation in the blood activity, so blood oxygenation, can be considered a reliable biomarker of a functional specialization within the brain, as showed by Reichel and et al. In 2010, and proposed recently in Nature Communication by Shock and Kojori. Now, what we know is that the complex resting state functional architecture of the brain supports his rich cognitive and behavioral repertoire, and the proper tool to investigate and describe this functional architecture can be found in complex networks and the theory behind. Now, how can we move from a bold signal as measured by fMRI to a complex network? Well, so typically you isolate the gray matter because you expect the brain expected to produce activity essentially in the gray matter, in the gray voxel, and you extract time series and calculate a similarity index between the two time series. Typically, you do that with the correlation coefficient. You try to move away from the problem of the sign by calculating the square or calculating the absolute value, or a distance according to the Euclidean distance according to the correlation coefficient. Anyway, at the end of the story, you put a weight on a link connecting two regions that are those that produce this two time series, and you do that for all the pairs of regions that you include in your investigation. For example, in this way, in this picture, we use the voxels of the gray matter, so we have 10,000 regions, of course. Then after a proper thresholding, you sparsify your matrix and define your network and try to investigate topological properties instead of statistics of co-activation or coherence. The problem is that the thresholding is there is not one way to threshold your network. You can use the statistics. Yes, you can say that you retain only ways that are significant, statistically significant in terms of the R. So P associating to a p-value, then you have to correct for multiple comparison the p-values associated to this correlation coefficient, or you can use topological methods. So, for example, you can ask for a small wordness of the network, so you fix the Erdős-Renyi entropy, or you ask for a balance between the cost and the efficiency of the transfer and information transfer within a network, as done by Enrico Fallani and Vito Latora, by optimizing the density according to the local and global efficiency of the network. In our approach, we didn't use any specific threshold. So what we did has been to progressively cut elements from the weakest to the strongest. So we sorted the weights of the network from the weakest to the strongest, and removed progressively these links, and watched what happened to the networks inducing a percolation process. Okay, and we did it on parcellation of the brain according to 116 areas, so it is not a voxel-based analysis. So we averaged the signal within these anatomical defined regions. And the curve that you see, the percolation curve, the blue one, has been obtained as the percolation curve of the average correlation matrix across 40 healthy subjects. The average has been done passing through the efficient transformation. And what is very interesting is that not only the slope of the experimental empirical percolation curves is different as compared to the randomized percolation curve, where the randomized percolation curve refers to the percolation process over the randomized matrix that comes from the experimental matrix. So we used a specific neural model that has been obtained thanks to the intuition of Rostamas, Andrea, and Andrea Gabrielli, who found the chance to define a symmetric, semi-definite positive matrix that has the same spectrum of the experimental matrix. And by rotating the experimental matrix, we did 500,000 replicas and averaged them, and we obtained this different slope. But what is interesting is the presence of these plateaus in the percolation curve that represents a range, ranges of resistance to disconnection. So during these ranges of threshold, the number of components that are reported is on the y-axis doesn't change. If you take a look at different plateaus along the threshold range, you can see that you can find an integration process of regions within the whole network. So you start from, if you look at the threshold scale from right to left, you find first the interaction between pairs of regions, then you start integrating the occipital cortex because we have the colored nodes according to their macro anatomical location. So you have occipital red, temporal green, parietal yellow, and frontal blue, and cerebellar pink. So you have a progressive integration of regions within the brain, and you can observe that the first region that tend to integrate in a resting state experiment is the occipital cortex. Is not a so surprising, because during the resting state experiment, the subject were asked to stay in the scanner with eyes closed, fixating a white cross on a black screen. So the essentially is the resting state and a properly resting state. There is a small amount of continuous stimulation. Another approach that we use to investigate the brain topology has been to retain only the strongest connections, so the strongest weights, the largest weight between pairs of nodes. And we obtain the maximum spanning forest, and for a healthy brain, the forest composed by communities or components that are mainly linear, okay, as opposed to the randomized counterpart, where you have much distribution of star-like branched trees. And another approach has been to retain the strongest connections between components of the forest. And we obtain the maximum spanning tree that produce this description of the fundamental backbone of the connectivity of the brain, where you have a portion that is highly segregated as the cerebellum, and a distribution of frontal regions and parietal region across the tree. It's a chain-like structure, as opposed to its randomized counterpart, which is a star-like structure. It's quite important, and we will see it in a few minutes, if you want to compare different trees one with the other. We did the same analysis on schizophrenia patients that are psychiatric patients with behavioral outcome like psychosis, hallucinations, so on and so forth. But we observed that the variability across subjects of connectivity, so of weights or correlations essentially, is much larger in patients than in healthy subjects. So we couldn't average matrices one with the other. We studied the groups subject by subject. You see that the distribution of the coefficient variation of the weights of the network in the two groups, red schizophrenia patients and blue healthy subjects, is different. It has been statistically tested with a Mann-Wilcoxon test. And we calculated the percolation curve. We found that the percolation curve were slightly different. The shadow, the colored shadow here around points, is the standard error of the average of the curves. So we didn't average matrices, but we averaged the curves. We lost a bit, so we smoothed a bit the curves. So we lost a bit the presence of the plateaus, but if you focus, some of them is still present. So here and here. What's the origin of this difference? We showed that we analyzed the distribution of the weights, and we found that there is a sensible reduction of the total strength of the connectivity within the brain in schizophrenia patients as compared to healthy subjects, but it's not enough to explain the different topologies. So we calculated degree, the average degree across subjects, region by regions. We ordered a plot here according to the variation of the degree along the threshold. So you have threshold and x-axis, and y and regions on y-axis. The color is the degree. And you can see that there is a different distribution of the degree variation in patients as respect healthy subjects. The degree of variation has been obtained as the square root of the summation of the difference of the degree of a region at a threshold minus the threshold before. We did also another observation. We calculated the size of the giant component in the network and ordered it according to when the here this on y-axis are subjects. So you have the giant component include all the regions in these areas and one region in these areas. And what you can see is that there is a sort of resistance to disconnection of the network in patient as compared to healthy subject. We calculated the area under the curve with a trapezoidal algorithm, and the difference of the area under the curve of two consecutive curves normalized to the largest one seems to be more resistant in schizophrenia, the disconnection, but at the end of the story, after a couple of changes of giant component size, the network crumbles, matching the size that you can obtain with from healthy subjects. We calculated the spanning forest for patients and subjects. It was not so much different. Also, the maximum spanning tree was similar. So, what we wondered was how can we quantify the difference between these two backbones? They are different at size, but not so different. So, what we have done has been to use an allometric approach. So, the idea is to take a node as a root and calculate two entities: the resource exchanged at the node I, so at the root, represented by the number of nodes forming the subtree of the root, including the root, and the total cost of transfer, calculated as the summation of the resource exchanged by each node of the subtree. And the relationship between A and C, resource and total cost, is an exponential relation, and eta is the allometric exponent. And eta can assume values between 2 and 1, with 2 associated to the linear structure, while close to 1 in a star-like structure. And we did it on our tree by considering each region of interest as a possible root, and the direction was obtained naturally by considering the root as the node as a root. So it's like if you pick up your network, your tree by the root you decide, and the gravity define in some way the direction, and you find as water flowing through the tree the possible directions and the possible subtrees. We did it for all the ROIs, and we obtained the first interesting thing is that the allometric exponent was 1.5, exactly between 2 and 1. So we are rightly describing a branched tree structure, but the problem was that a schizophrenia patient, a healthy subject, was essentially reported essentially the same allometric exponent. On the x-axis you have ROI roots, and the shadows are the standard errors. But we wondered if what happens if you remove the maximum spanning tree from your network, and then you consider the maximum spanning tree of the remaining network, so the second order maximum spanning tree. We did it and calculated the exponential exponent, the allometric exponent. We found that healthy subjects in blue seem to reduce the allometric exponent more than schizophrenia patients. So we did it to a second order, third order maximum spanning tree, and the difference increase, and so for the fourth, the two curves separate. So it seems that, so we interpreted that as the process during which several equivalent maximum spanning tree are present in schizophrenia patient in terms of connectivity and organization of connectivity, as opposed to the healthy subjects that present just one main maximum spanning tree. And this is maybe a compensatory effect, because we know that the reshuffling and the of weights induced by the pathology tends to destroy the modular structure of the hierarchical modular structure of the brain functional connectivity, as showed by the group of Professor Bifone, due to the decrease of connectivity strength, as previously demonstrated by the group of Professor Bassett. And we think that the overall organization of the schizophrenia brain is essentially characterized by this tentative compensatory process that try to make the brain functioning, even if there's unbalanced reshuffling or weights of connections of functional coherence is induced by the pathology. So to sum up, we demonstrated percolation highlights intrinsic hierarchical configuration of the brain, and the maximum spanning tree revealed the intrinsic backbone of the functional network, and together they accounts and support this connectivity hypothesis of schizophrenia in terms of the that can explain behavioral alteration in neuropsychiatric patients. So let me thank the authors of this research, Sana Mastrandrea from IMT, Andrea Gabrielli from Roma 3, Guido Caldarelli e Ca' Foscari, and Fabrizio Piras e Gianfranco Spalletta for Santa Lucia Foundation, and you for your attention.
Thank you, Tommaso, for the talk. There is time for some questions. Please, Egidio. So the microphone is here.
So thank you, very interesting, very stimulating. And I wonder whether you have an idea of what the subnetworks do in this play. So basically, if you can extract the specific change or role, put it in the other way, of a subnetwork in regulating the connectivity.
Yeah, we are studying this in a side project about the. We would like to go behind the maximum spanning tree and try to explain these differences according to the subnet that sustain these changes. What we know is that if you look at the connectivity and the modular and the communities that can be observed by community detection algorithms on this network. For example, somatosensory community is destroyed by the pathology, and the correlation between the outcomes, behavioral outcomes, and the level of the disaggregation of this module is very high. That has been showed by Bourdieu et al. On neuroimage.
Yeah, I understand this clearly one of the developments, and I wonder, you mentioned the cerebellum a couple of times, and I wonder whether you have a specific indication about the subnetworks that the cerebellum is doing with the cerebral cortex.
Absolutely, we have some insight about the cerebellum that in the investigation of the Wool network, we found that cerebellum is a sort of bridge between the two hemispheres, the different regions of the two hemispheres, in terms of network. We did it by a method that, with Andrea, Miguel Berganza, and our student Francesca Santucci are doing in regulation, in the regularization of the correlation matrix and the inversion to calculate the partial correlation. And by percolating that matrix, we find that the cerebellum is a central point of communication between the two lobes and the different regions of the two lobes, so it has a high betweenness. But during the pathology, for the pathology, the cerebellum subnetwork is disgregated. Absolutely, yeah. And it is the same in autism. We are doing. I'm doing the same analysis with the ISI in Turin on autism data, and we found quite the same results.
Quite interesting. Thank you.
The second question, Maurizio, please.
I'm wondering whether you considered also some inhomogeneities in time. I mean, are you sure that the subject is not having a nap, or I didn't get exactly whether you are providing some stimulation or if you so he has a blank screen.
Yeah, they watched they only watched a black screen with a white cross. So the time series we got from the both signal has not been investigated in terms of the inhomogeneities of time dispersion at different time scales. But I think that for this temporal resolution, that those inhomogeneities are quite complicated to find. I think that you should improve temporal resolution and measure them with, for example, neurophysiological experiments where you have a lot of points and you can do a proper analysis of different time scales.
Another last question, Stefano, did you want?
Yeah, dealing with patients is always difficult, and I'm wondering, given the fact that schizophrenic are supposed to be a very heterogeneous group, if you...
Please mute for people participating from remote, please mute the microphone.
Yeah, I wonder if you considered this fact, and also probably more basic, what is a resting state for a schizophrenic patient? Is it possible really to have a resting state or patient staying still for 15 minutes, given that they are schizophrenic?
Okay, this is a very interesting question. That subjects have been selected according to specific recruitment rules by the medical doctor. So we try to put together a sample, a cohort with behavioral homogeneous patients. The inter-subject variability has been the problem that we had in calculating all the stuff, and we had to move from the average correlation matrix to the single subject correlation matrix. As far as the resting state of psychiatric patients, yes, this is we cannot properly describe them in terms of what is the resting state of these patients. We assume that while doing something like staying a scanner, fixating across a white cross, unmedicated subjects could return the same bold signal and coherence across the brain of bold signal. But I agree with you that the resting state of a psychiatric patient is different as compared to the healthy subject. The problem is that you have to enroll patients that must be medicated, collaborative, and try to find possible homogeneous results.
Very, very quickly, my question was, but you got the point. If you're if you don't know if the behavior is exactly the same, then it's difficult to make points on the differences.
Yeah, but they have been widely tested by the hospital We enrolled this cohort in the hospital by psychologists and medical doctors. After all the battery of cognitive examination, we selected 40 subjects out of 150, 200 subjects. So we have also the reason for this small number of subjects is due to this reason, and this is the problem for large repository of patients that must be carefully investigated.
Okay, thank you, Tommaso. Thank you again. We move